Good evening, good day to everyone, and welcome to our IVF webinar. Once again, you can see our guest, Elena is back. She was here with us already on Tuesday, and now we do have another interesting topic, and she has a wonderful and definitely interesting presentation with her tonight as well. And for those who weren't able to join us on Tuesday, let me just mention that Elena Lloret, she is the Biomedical Scientist at iGLS, which is a Reproductive Genetics Laboratory of IVF Life Group, and she will, of course, tell you a little bit more about it as well. But as you can see today, we definitely have a very interesting topic on NK cells, and I can always see lots of questions whenever we are mentioning NK cells. So I'm really looking forward to this session. And of course, remember that if you have any questions, you will be able to ask them, and Elena is right here to help you out with those questions. We will have a Q&A session right after the presentation, so don't hesitate, just put those in the chat section so that Elena can help you out with those. And welcome back, Elena. How are you feeling tonight? Hope you are feeling very nice and ready to for this session as well.
Yeah, thank you again for the invitation. I'm really looking forward to give this presentation. I hope everyone enjoys it. And yeah, I'm more than happy to answer any questions at the end, as you were saying, Caroline. So yeah, I'm looking forward to this presentation.
Wonderful. Thank you so much for joining us here once again. And now I guess let's begin with our presentation first, okay? Thank you.
Thank you. So yeah, let's start. As Caroline was saying, my name is Elena Lloret. I'm a biomedical scientist at iGLS, which is the Genetics and Immunology in-house lab of the IVF Life Group. And yeah, we're here today to talk about natural killer cells at IVF, which is quite a controversial topic that I'm sure everyone would be really interested to listen about tonight. And yeah, I hope you like this presentation. So yeah, let's start. Let's have a look at the plan for tonight's session. At the beginning, I always like to give the context of the topic. I'd like to introduce you to the immunological context of implantation, and therefore why natural killer cells are so important for this process. So to begin with, we will look at the embryo, and I'll try to explain how the embryo is an exchange body for the woman's immune system, how it's detected, and how the body reacts to it. Then we're going to look at the endometrium, which is, is a crucial structure for implantation. So, and as well as constantly changing, which is really a challenging situation. And then we're going to look at the immunological challenges that all these circumstances have. And then we'll go more in depth into the topic, which I'm sure you all want to hear about. So yeah, we'll be looking at natural killer cells. We're going to look at the different phenotypes that we can find throughout the body. And definitely we're going to look into the specific phenotype that is found at the uterus. Then we're going to look at the functions of these cells and how they are so crucial for implantation and early stages of pregnancy. And then we're going to look even more in depth into the maternal and fetal interactions through the molecules that are implicated in these crosstalk. So let's start. Yeah, so as I was saying, an embryo, as you know, it has half of the genetic material from the mother and half of the genetic material from the father. So that means genes, they get expressed, so we also express proteins in our cells. So that's what happens in the embryo. The embryo expresses half of these proteins from the mother and the other half from the father. Therefore, when the mother gets in touch with this embryo for the first time, it will be seen or be detected as a foreign body. So this is known as a semiallogenic organism because it's got half of the genetic material from the mother, which will be shared with the mother, and the other one from the father. So therefore, it's possible to develop an immune response. However, cells at the extravillous trophoblast, which is the outer membrane of the embryo, of the trophoblast, present specific molecules that limit the classic recognition, and therefore this reduces the risk of rejection. But however, it still implies a really important immunological challenge as the embryo invades the lining of the uterus, which is known as the endometrium. I'm sure you've heard about this term, and even that invasion implies a change in the cellular structure of the cells in the endometrium, which is known as desivillation. So let's see what's going on here. As we were saying, one of the main players here is the endometrium. So as you know, the endometrium is constantly changing structures because we've got a menstrual cycle, and because of the hormones, these influence the cellular level and the structure of this tissue that is changing throughout the cycle. But as you may know, the endometrium is a mucosa and is an initial barrier for the body. So it's really important to understand the mechanisms in these structure, because it has to protect the body from the invasion of other pathogen organisms. However, it also has to be tolerant towards the embryo that is going to be trying to implant. So yeah. We're going to look at the different populations of the immunological cells that are at the endometrium. As I was saying, this is a really, really complex situation, and therefore it requires some specific mechanisms. As been suggested, then there are some specific homing mechanisms that make all these immune cells specific. They present a specific phenotype that is more tolerant towards the invasion of the embryo. Such as, for example, the macrophages. Macrophages present a different phenotype known as M2 that stimulate endometrium receptivity, as well as the lutein body formation that will then lead to the production of hormones that will allow the development of pregnancy. As well, promotes the uterine vascularization, which is crucial. I cannot emphasize it enough for placentation. It will, as we will see later on in the presentation, it will be really important to have an adequate vascularization in order to have an adequate implantation and early stages of pregnancies. Now we're going to look at the T lymphocytes. T lymphocytes here are very different. We've got very different populations that are either more pro-inflammatory or more tolerant. However, they present a different a variant of their receptors and their membrane, which is allowing this recognition to be the recognition with the embryo to be less immunogenic. But yeah, we do require this inflammation at certain stages of implantation, for example, at the beginning, because we require this vascularization to occur. Therefore, we do need the Th1 and Th17 cells that promote pro-inflammatory cytokines production and will allow implantation. However, then in later stages of pregnancy, we will require the activity of Th2 cells and regulatory cells that will allow the establishment of tolerance in the body. But yeah, we were not here to talk about all these. We were here to talk about natural killers. So yeah, let's have a look at the natural killers itself. As you may think, natural killers, that's a pretty aggressive name. So how are these so important for pregnancy? Well, there are different phenotypes of natural killers throughout the body. For example, in the peripheral blood, we can find two specific phenotypes, but mainly one that is more cytotoxic, is an initial barrier towards any infection, any pathogen that may attack the body, specifically to viruses. However, there's still a 10% of these population in the peripheral blood that presents a different phenotype. It's more tolerant. It's got different functions. It's more focused on cytokine production and therefore regulation. So the phenotype that we find at the uterus is quite similar to these 10%. It's focused on regulation. It produces lots of cytokines. It produces angiogenic factors that will be crucial for vascularization, and yeah, they do have a reduced cytotoxicity. For those who don't know what cytotoxicity means, they are not so aggressive towards other cells. However, it's important to mention here that these phenotype is also dependent on the context. As you can see there, is dependent on interleukin-2, which is a pro-inflammatory interleukin that could be found at the endometrium. So I want to emphasize here that it's not that we have different players that are playing their own game. It's a really complex situation that we have to understand on the whole of it, like not just independently. So yeah, natural killers, in theory, at the uterus, they exhibit this phenotype, but sometimes, depending on the patient or on the situation, if the patient has suffered an infection or many other circumstances, this phenotype may vary a little bit. So it's really important to look at every case independently. So I may say something here that may not be useful for all of you, but may be useful for some. So definitely look at the cases independently. Okay, so now we're going to have a look at the evolution of the natural killers throughout the cycle. And then we're going to try to understand from these presence their importance at the site of implantation. So at the beginning of the cycle, there's a low level of decidual natural killers or uterine natural killers. Both names can be used because they are not so important at that point. However, when ovulation occurs, it's been seen that the structure of these natural killers change. They become larger, they've got more granulites that have been thought to participate on regulation and secretion of androgenic factors that we will need later on for placentation. And then there's a great peak of these natural killers at the window of implantation. Therefore, they have been thought to be really important for this process. Then, if in the event of pregnancy, the natural killers populations remain high, accounting to up to 70% of the whole leukocyte population of the uterus, which is a large number compared to their proportion in peripheral blood. They usually don't account for that much of the leukocyte population in the peripheral blood. However, they become crucial at the uterus when pregnancy, in early stages of pregnancy. And then if there's no implantation and if there's no pregnancy, then the hormone levels go down, and then these natural killers go to apoptosis, which is controlled cell death. So the cycle begins again. So yeah, now we're going to look at the relevance of these natural killers and placentation itself. As you may know, placentation is the moment where the embryo invades the uterus, and then there's an interaction between the maternal blood vessels and the embryo, and then they establish all this structure. As you can see here in the diagrams, where the blood vessels invade the area around the embryo that will lead to the development of the placenta. It's really important. There's two main characteristics here that are really important in order to establish an adequate placenta. That are the spiral arteries remodulation, and then also the connection, the crosstalk between the embryo and the maternal immune system is really important. We don't want an excessive invasion of the uterus, and we don't want an excessive rejection of these embryo. So the crosstalk between these two pieces of the game are really important. So how these interactions occur? Well, the main players here are, well, as we can see here, we've got the mother and the embryo. These are our two players at the moment. Then specifically, we're going to be looking at the extravillous trophoblast cells, which are the cells around the trophoblast, the cells in the more external lining of the blastocyst. And then we're going to be looking at the natural killer cells. So specifically, from the natural killer cells, we will be looking at certain type of receptors that is known as KIR receptors. And from the extravillous trophoblast cells, we're going to be looking at the human-like antigen C. These interactions are going to be crucial for determine how placentation occurs, how natural killers react towards the invasion of the embryo, how they secrete androgenic factors, how these vascular remodeling occurs. So these interactions is definitely important. So yeah, let's have a look at the key receptors. So key receptors are formed by 16 genes that can be either translated into activating proteins, which are those that have the S in their nomenclature, and those that have the L in the nomenclature, which implies a long tail and a short tail. So the long tail express inhibitory functions towards the natural killer cell, and the ones having small tails express activating functions towards the natural killer. So with these 16 genes, there has been created two haplotypes have been created that are KIR A or KIR AA or KIR BX. Okay, so the KIR AA is predominantly inhibitory, whereas the KIR BX is predominantly activating. So as we were saying before, the activation of natural killer cells is really, really important for an adequate placentation. Because if we don't have fully functioning natural killers, then we won't have enough androgenic factors or enough vascular remodeling, or the relationship or the crosstalk between the embryo and the maternal immune system won't be done properly. Therefore, we won't have a successful pregnancy. So as I was saying, this is really important. So how these occurs, this interaction between the KIR receptors and the extravillous trophoblast, where these occurs throughout some molecules known as a human-like antigen. As you can see in the diagram, there are different ligands to these receptors. However, here we're going to be looking specifically to the HLA-C molecules, because they will determine the activation of natural killers. So yeah, let's have a look. As we were saying, we can have different phenotypes of different genotypes of the key receptor that are going to be mainly separated into two groups: KIR AA, which are predominantly inhibitory, and KIR BX that are predominantly activating. So because we've got the activating one, then these in the cases where the mom is got a KIR BX genotype, then we usually don't have problems with natural killer cell activations, because they usually lead to an appropriate natural killer activation. Whereas we've got the problem with the natural killer cells that present a KIR AA genotype. Okay, so let's have a look. Also, it's really important to mention that we've got two different alleles for the HLA-C. We can have either HLA-C1 or HLA-C2. This leads to three different possibilities, which are homogeneous for C1, which is C1, C1, heterogeneous C1, C2, or homogeneous for C2. Okay, so because the human body reacts towards the foreign molecules, if we've got a larger load of C2 alleles in the embryo than there is in the mother, then the natural killer cells won't be activated. There will be an inhibitory response, which leads to reduced androgenic factors production and reduced extravillous trophoblast invasion, and therefore not an adequate placentation and implantation. However, if we've got the same load or even a lower load of HLA-2 alleles in the embryo than there is in the mother, then it should be okay implantation. However, it is recommended to do a single embryo transfer, because if we do a double embryo transfer, these loads may increase the load that is found at the maternal system. Therefore, there may be inhibitory functions as well. I hope this is clear, but if you've got any questions, I'll be more than happy to answer them later. Yeah, as I wanted to emphasize as well, that why is it C2 so important? Well, C2 alleles are specific ligands to specific inhibitory receptors, therefore that are found mainly in KIR-AA genotype. Therefore, the greater loads will imply a greater binding between these ligand and the receptor, and therefore a greater inhibition. But yeah, as I was saying, if you've got any doubts, I'll be more than happy to answer them later. Yeah, let's have a look at these HLA-C genotype. I don't know if you've got some knowledge about genetics, but mainly going quickly into it. It's important to differentiate between the genotype of the mother and the father and the embryo, because as I was saying at the beginning, the embryo will be a mixture between these two. So if we follow the genetics laws of Mendel, then we will see that if we've got a heterogeneous mother with a heterogeneous father, then we will have 25% of possibilities that the embryo is homogeneous for C1, and 50% of probability that we've got a heterogeneous embryo, and then 25% possibilities that we've got C2, so therefore homogeneous for C2 embryo. Then we've got two homogeneous fathers for C1. Then all the embryos produced by these couple, they will be homogeneous for C1. And then if we've got a mixture, we've got a C1 homogeneous mother and C2 homogeneous father, then we will have heterogeneous embryos. This is important because later on we're going to be looking at the recommendations for the treatments, and it's important to understand how sometimes we do genotype both the mother and the father, but we don't really think that the embryo may have different genotypes, may not be exactly the same as the mother or the father. So I wanted to give you these kind of knowledge so you've got some perspective. Okay, so yeah, as I was saying, let's have a look at KIR and HLA-C combinations. So as we said earlier on, mainly we've got these problems or these low activation situations of the natural killer cells in the cases of KIR-AA mothers. So when they present a KIR-AA genotype, they usually have these low activation. However, we're going to be looking at these because it doesn't happen always. As we were saying, the HLA-C concentration or the expression of these HLA-C molecule by the embryo will definitely determine the reaction of the natural killer cells at the endometrium. So I don't want you to just get tested and see that you're KIR-AA and think that you're never going to be able to conceive, because that's not the situation. We just have to look at the different possibilities and try to organize it or combine it so these implantation can occur properly. So yeah, as I was saying, if we have a look here at the KIR-AA mother, well, this is the case of own ovocytes and the couple sperm. So this would be just the couple. If we have a look at the KIR-AA mother, when we've got in C1, so homogeneous HLA-C genotype, then we have to look at the HLA-C load of the sperm, because if it's greater than the presence of these alleles at the mother, then we're going to have some issues. So we'll have to be really conscious of these. Then if we've got a heterogeneous mother, then we also have to, here the proportion is not so high, but still we have to be definitely looking into it talk to our gynecologist and see what options they can give us, because sometimes using a sperm donor is a good possibility. If not, some treatments are available as well. Then here, in this case, I think this is quite interesting. I hope you understand these clearly. So for KIR-AA women that have a homogeneous haplotype, homogeneous genotype of C2, when we have a C1 sperm cell, then we won't have a problem here, because the presence of C2 allele in the mother is greater than there is in the father. Therefore, the embryo 1% greater concentration of these molecules. However, if we're looking at a C2 sperm cell, then there is possibility that these workload is higher if we do have a double embryo transfer. Therefore, a single embryo transfer is recommended. And yeah, as I was saying, for KIR, the same thing happens when we've got C2 and C2 sperm cell. And as I was saying, for BX, we don't have that many problems, because these genotype is usually activated, and therefore the activation of these receptors will lead to an appropriate natural killer cellular functioning and cytokine production that will lead to an adequate placentation. So sometimes we do require a sperm donor. So it's basically the same situation here than we had earlier on, because at the end of the day, the sperm is not involved in the maternal immune system. So it's not as crucial to determine the donor. However, it is crucial to determine the donor. We need to have a lower load of C2 and molecules present in the embryo than there is in the mother. It is even more interesting to look at the egg donor situation, because what we're looking for is to have the same genotype that we have in the gestating mother, because then we will ensure that we don't have a greater concentration of these HLA-C2 molecules in the embryo than there is in the mother. And yeah, to finish off this presentation, I'd like to say that at iGLS, we do have the possibility to access to these tests, both tier and HLA-C genotyping, which is really useful for cases where we don't really understand the cause of infertility, or we've had different situations of abortions or even implantation failures. So it is interesting to sometimes have a look at the immunological context to see what is going on there. And especially in this topic, we also do an immunological map of the endometrium, known as EMAP, and it is definitely interesting to look at these tests, because as I was saying earlier on, immunology is really complex subject. Is very global. We have to look at it from many different perspectives, and sometimes the situation seems really very clear, but then it is not, because we're not looking at the whole picture. So well, these tests allow us to have a look even more into the whole picture than we do look at sometimes. So yeah, if you're interested, you've got the email there, so we'll be more than happy to help you out. And yeah, this is all my presentation for today. And right now, I'm sure I can see there are lots of questions, so I'll be more than happy to try to answer them. And yeah, I hope you like the presentation, and thank you very much for your attention.
And thank you very much for that very interesting presentation. Lots and lots of details. This is never an easy topic, but in my opinion, you definitely did great. And it was really nice to see all those details and how you explain things. I think you made it quite clear as well. Elena, thank you so much. But as you can see, lots of questions are right here coming in. So let's get to them. Okay, let's get to those questions. I'm sure it's going to be an interesting session tonight for sure. Okay, let's have a look. The first one is here. From my biopsy, I can see a value Th1 cell within range if this considered in percent, but high when considered as cell numbers. Is it possible? What does it mean?
Well, thank you for your question, GB. Sorry, GB. Okay, so what I'm getting for your question is that the results is in a percentage, but no. Okay, so what I'm assuming here, you're talking about the ratio, because Th2, as I was saying, from the TH cells, we've got two different responses, which is Th1 and Th2. So Th1 are more pro-inflammatory cells. They secrete pro-inflammatory cytokines, and they do have a more pro-inflammatory function, whereas the Th2, they are more regulatory and more tolerant. So what I'm guessing is that you have an adequate ratio between these two populations, the Th1 and Th2 cells. That's what I'm understanding from the percentage. And however, you do have an elevated number of Th1 cells. Well, as I was saying, well, it is possible. Yeah, definitely, it is possible. And what does it mean? Well, what I can give you, the answer that I can give you right now, without looking at the whole study, is that it may imply that you've got a more pro-inflammatory response, but as I said earlier on, this is a really dynamic situation. So it may be because of the moment of the cycle where the biopsy was taken, or it may be because you have had like an infection or something, or even it may be due to some medications that sometimes we take, but we're not really conscious of their effects. So I hope these answer your questions, but yeah, definitely I encourage you to get in touch with an immunologist that will look at your case more into depth. As I said earlier on, if you want to get in touch with us, you have the email there, and we'll be more than happy to try to answer those questions. But I can't really give you a personal answer without looking at the whole picture. Yeah.
That's of course, that makes sense. Of course, thanks so much for your question, and thank you for your answer. Let's have a look. More questions are coming in. So, how does mother is affected? I guess HLA-C in a double egg donation program.
Okay. Here we, as I said, we do have to. So. The natural killer cells are going to detect as foreign organisms or cells that do not present the same amount of HLA-C that they do present in the receptive mother. So if we do have a double egg donation, we do have to be really careful about the HLA-C status. So ideally, it has the same, it presents, the egg donor presents the same HLA-C genotype than the receptor, because then this will ensure that we don't have an excessive amount of HLA-C2 molecules. But yeah, thank you very much for your question, Carla. It's a really interesting one.
So much indeed, Carla. And of course, let's have a look. More questions are coming in. So, is Chicago test reliable identification of elevated NK cells that are affecting implantation, or is biopsy required to know for sure?
Well, that's a really interesting question as well. Thank you, Monica. From my experience, I do recommend a biopsy. As I said before, we do need to look at the situation at the moment and the site of implantation. Especially, we also have to look, the biopsy should be taken during the window of implantation, because as I explained there in the presentation, the endometrium is a dynamic structure that will be changing. Their populations will be changing throughout the cycle. So I think, from my experience, again, I encourage you to get a biopsy.
Again, thanks so much indeed for yet another interesting question. And next question is ready. What are the risks of having immune treatment in relation to NK cells and implantation failures? Will these treatments affect my immune system long term?
Well, thank you very much for your question, Monica, again. That's a very good question, because definitely immune treatments we have to be really careful about them. The immune system is really complex. It's definitely something to be really careful about. I will not play with it. So because of that, I think, as I said earlier, I think it's crucial to have a look at the personal situation. However, there are some treatments, for example, as I was saying, for implantation, sometimes there's not enough activation of natural killer cells, and this is affecting the vascular remodeling. So sometimes, if we do take medicine, I'm not encouraging anyone to do these, but there are some medicines that are not immune treatments, that are not immune modulatory drugs or treatments that may help as well, such as, for example, aspirin, because these helps to remodulate the vascularization of the uterus. So it may be helpful, and sometimes we don't need to have an immune therapy that, as you were saying, of course, is complex and and difficult to consider. So I definitely, first of all, I definitely encourage you to get in touch with a specialist, an immunologist, and a doctor. Together, I'm sure they can see your case specifically, because there are so many things that sometimes we're not aware of that we may suffer, and it may seem like a silly detail, but I'm sure they'll be able to detect the importance of these details, such as like your family history or like elementary habits, and that may be affecting all these situations. So I definitely encourage you to get in touch with a specialist, an immunologist, and have a careful look at these immune treatments that of course are difficult to manage sometimes.
Wonderful. Again, thank you so much for your follow-up question, and definitely interesting answer as well. And let's have a look. Okay, so what is the test to get KIR and HLA couple combination?
Well, I'm assuming you're asking about the test that I put up. Well, the test basically what it does is like sequence the DNA from both the mother and the father, and then we genotype for KIR and HLA-C. So then we can see what situation we have is basically the pictures that I put up, the tables that I put up. So we will see that, for example, the mother is BX, then maybe we don't need to go more in depth here, because, well, as we said, sorry, I don't know, a question came up. I'm not sure if it's like a follow-up question. Yeah, it's for the test, it's a blood test. For the KIR HLA-C, it's a blood test for both the mother and the father. We just need a blood test, because these KIR and HLA-C receptors molecules are expressed in like all the cells of your body. But in these contexts, we're looking at specifically for the interaction in the implantation site. So yeah, basically take a blood sample from the mother and the father. As I was saying, it'd be interesting to test the mother earlier to see if there's if she has a KIR AA or KIR BX. Then if we do have a KIR BX situation, well, maybe this is not the reason why we're having implantation failures or other pathologies. However, if we do have a KIR AA, then we may also want to look more in detail into the HLA-C status of both the mother and the father, and then see the situation, and therefore be able to see if there's actually a problem with that combination, or is just something to take out of the picture. So yeah, it's just a blood sample.
Thanks so much for the clarification. And yeah, sorry, that was a follow-up. I just wanted
to... No, thank you. Yeah.
And let's have a look. Another question is right here. So do you believe that this is why stats are 40% M male, I guess, and 40% F in fertility, with 20% of mix towards outcome?
Well, thank you, Derek, for this question. Well, from what I'm guessing is that you're asking if the immunological infertility or problems are the cause of these 20%, 20% of the pregnancy failures. Well, definitely, if that's your question, definitely immunological issues are a big part of infertility. And as I was saying earlier, sometimes we just look at the quality of the embryo, which is really important as well, or the window of implantation. They are really important factors, but there are some cases where these factors are controlled. The embryo is really good. We've done PGS, we've done tests to detect the window of implantation of the endometrium, but we're losing a big part of the whole picture, which is the immunological context of the implantation site, that is at the end of the day the maternal uterus. So I definitely believe that immunology has a big part in these pregnancy failures. So I recommend everyone that is suffering from these unknown infertilities to look more in depth. Yeah, thank you.
Excellent. As you can see, thank you from Derek. And let's have a look. Next question. If you can clarify, is the test checking this the ER map at iGLS or the IMAP?
Yeah, thank you for the question, Christina. Yeah, we do have both. So the AIR map is the endometrial receptivity map. So that determines when the window of implantation is placed, because as you may know, different women have different window of implantation. So it may be when they 5 days after the progesterone is going high, or 7 days after ovulation, or for other women it may be shorter, it may be longer, it may be we have many different situations. So that's the AIR map test that determines where the window of implantation is placed. However, I was talking about the EMAP test, which is the immunological map. And here we're looking at different populations. We're looking at T cells, we're looking at cytotoxic cells, which I didn't mention here, but they basically are cytotoxic. So they attack the cells that are in the uterus. They are more aggressive. So we want to control the percentage of these cells. Then we also look at the TH cells, as we mentioned earlier in the first or second question. It's important to look at the ratio between TH1 and TH2 because the TH1 cells are more pro-inflammatory, whereas the TH2 are more tolerant. So we require different situation at different moments in the implantation and cycle at the moments in the cycle. We're also looking at B lymphocyte cells that I think I just remember that I didn't talk about. Well, B cells should not be present in the endometrium really, but sometimes we do have a specific phenotype that is known as B2 that are thought to be helpful for implantation, and then we also have another phenotype which is known as B1A, which indicates some kind of autoimmunity. So we detect all these populations. This is quite wide range of populations that we detect. So, well, of course, we look at the natural killer cells, and yeah, so it's basically given us a piece of the whole picture. It gives us some information about what's going on at the endometrium, looking at the most important populations that will get involved in implantation. So yeah, sorry, these are the two differences. One, AIR map is endometrial receptivity, and EMAP is immunological map.
And thank you so much for the clarification. I think it's now very, very understandable. So thanks a lot. Let's have a look. Another question is here. From my biopsy, I have high NK and low NKT. How is it possible? What is the difference between these two markers?
Okay, so yeah, NKT are in a specific population that are between natural killer cells and cytotoxic cells. So as I was saying, well, of course, we have like a wide range of phenotypes here in specific cells. So these could happen because it's in a specific, it's kind of like an in-between population. These could happen, and as I was saying, I cannot give you like a proper diagnosis or anything like that, because I have to look at the whole picture. But natural killer cells, natural killer cells, uterine natural killer cells, those are the ones that have a more regulatory profile, whereas NKT cells have a more cytotoxic profile. So this is, of course, 100% possible because, yeah, as they are different populations. However, yeah, we'll be more than happy to look in depth to your case if you're interested.
Again, thanks a lot, of course. And another interesting question. Thank you for explaining. And one more question is also here. My NK cells came back high through blood work. Would you recommend I repeat with biopsy? I had intralipid therapy 2 times and then just had a failed transfer of 3AA day 5 embryo.
Yeah, so I don't know if you remember. Well, thank you very much for your question, Brittany. In one of the first slides, I was talking about natural killers. There are different phenotypes of natural killer cells, and the type of these cells between the blood and the uterus are completely different. We did a study looking at them, and they are different. So I 100% encourage you to have a biopsy. Yeah, because maybe sometimes the natural killer cells at the peripheral blood levels are higher. Sometimes the context at the uterus is different. So I 100% encourage you to do a biopsy. Yes. Hope to answer your question.
Thank you so much for that advice as well, of course. I think it does. But of course, I always repeat that if you would like to add anything, you know what to do. And let's have a look. Next question is also ready. Do you have any data on LIT treatment before embryo transfer?
Well, I'm not sure. Well, thank you for your question. I'm not sure what do you mean with LIT treatment. I don't know if you can give us more.
If you can perhaps clarify, we will be happy to go back to this question, okay? Because I'm not sure here either, I'm afraid. So
yeah, sorry.
If you can just let us know, I will go back to this specific question.
Thank you for the question anyways.
Of course, as always. So thanks a lot. And let's have a look. Brittany has added one more question. Would you recommend Levunex in combination with aspirin, Benadryl, Pepcid, and intralipid therapy? I was on all of this prior to my failed cycle. I can't help but think it was too much.
Well, thank you for this question, Brittany. As I was saying earlier, of course, I cannot recommend anything just by looking at the information now. We'll have to have a look more into that. But yeah, sometimes immunological treatments are complex, and combination of them is difficult to manage. So I definitely ask for an advice here to an expert that can look at the case more into depth. I'm afraid I cannot give you a complete answer here because, of course, I don't know the whole situation. But yeah, as I said earlier, sorry, but we'll be happy to look at them if you want to get in touch with us. Yeah, please do. But right now, I cannot give you a appropriate answer. Sorry.
Thank you still, of course, right here. And I just want to go back to the previous question. Okay, I'm not sure if you're familiar. I just checked. I needed to check. This is LIT, is lymphocyte immunization therapy.
Lymphocyte immunization therapy.
Exactly.
I'm sorry. I'm not familiar with these techniques, so I can't.
Okay.
Sorry, I can't really.
Not a problem at all. Of course, I just wanted to check it to see if it will help. But in such case, it's fine. Remember that, of course, we are focusing here on NK cells, right? So, thanks so much indeed.
Question, anyways, I'll have a look.
Yeah, definitely interesting, right? Something new for you as well. All right, excellent. Thanks so much. And let's have a look. Next question is, what can be the causes of the embryo implantation not resulting in a pregnancy if both ERMAP and EMAP were performed with no risk identified regarding NK cells?
Well, that's a very good question, Christina. It seems that you're familiar to the test. as I was saying, is difficult to predict. I don't know the quality of the embryos. I'm not sure if then the recommendations were followed. Then, of course, the EMAP test comes with some treatment suggestions. I'm assuming they were followed or not, but definitely, well, if you are already in touch with iGLS, because I'm assuming you are, definitely get in touch again with us, because it'll be really interesting to look at the case more in depth. I'm assuming, well, maybe the embryo quality that you didn't mention wasn't too high. This sometimes happen. I'm not sure if PGS was performed. If sometimes, well, embryos that have genetic aneuploidies and these defects, 5pp, well, that's a pretty good quality embryo. Yeah, as I was saying, sometimes aneuploidies can be, aneuploidies are a great cause. Yes, PGS was from as well. Well, I can't really give you an answer right now. I'm sorry to hear that this didn't work. But yeah, definitely get in touch with us again, because we'll be what can be the causes of embryo implantation. Oh, okay, this is the same question. Yeah, so yeah, definitely get in touch with us, because yeah, I'm assuming that maybe a treatment didn't work properly or wasn't done right. So yeah, the only thing I can tell you is get in touch with us and we will look at the case more in depth. Yeah.
Still fine, of course. Remember that you will be able to get in touch with Elena, and I'm sure whole team will be happy to help you out. Yeah, sometimes it's needed, more details, of course, are needed. So I'm sure they will be able to help you out. Will do. Thanks. Thanks so much indeed. Okay, next question is here. If steroids would be prescribed, should they be started along with estrogens for frozen embryo transfer preparation, or even from 1 week before transfer?
Well, this has to be determined by the doctor. Of course, I cannot prescribe any medicine. But yeah, I think if I'm not wrong, they should be started along with the transfer preparation, or even earlier. Sometimes we do need, because as I said, immunology is complex and difficult sometimes. So sometimes we do need a period of time where we have to wait to see how all these treatments are affecting, and sometimes even like repeat the test. And sometimes with blood tests, we can see how the levels of these cytokines production is working. But yeah, sometimes we do require more time to see if the treatment is really helping, or we do need to change the treatment. So yeah, I hope these answer your question.
Again, of course, for the interesting question and your help. Okay, if you can clarify this one, does EMAP have a specific window for being performed during a cycle timeline?
Yeah, well, it's recommended to do it at the window of implantation, as I was saying, because, well, as you can see at the presentation, for example, the natural killer cells can be really low at the beginning of the cycle, and this doesn't imply that at the window of implantation we will also have low levels of natural killer cells. It's just that it's not done at the moment where they have to be high. So yeah, definitely, I definitely encourage to take this biopsy during the window of implantation. I hope this helped, Christina.
Once again for your question, Elena. There are like few questions left. We will be slowly finishing. So if you have anything else that you would like to ask, go ahead.
To answer.
Go ahead and type those in, of course. And let's have a look. What are the risks and benefits of intralipid infusion for cases where embryo implantation did not result in a pregnancy but not in miscarriage either.
Sorry, I'm trying to understand the last bit. So where the embryo implantation did not result in pregnancy, but did not miscarriage either. So basically, implantation did not occur. So it would be a failure of implantation. Well, these cases should be taken as like in different. We do take different cases in different situations. Intralipid, as far as I'm aware, intralipid does help to promote natural killers functioning and development. So the risks, to be honest, I'm not 100% aware of all the risks because, well, intralipid is not as strong, let's call it like that, as other immunological treatments. However, it does modulate as well, so we have to be careful with it as well. I'm not saying that we should all take it because, we have to be careful with it. But I'm not fully sure of the risk. I do know that it's been reported to help to establish implantation, but I am not fully aware of the risks.
Well, thanks so much again. And there was one more follow-up from Christina from the previous question, because you've mentioned about the window when it should be done. So thanks, it was done outside that window.
Okay, I'm glad then. This makes a lot of sense then. So I encourage to do it again at the window of implantation.
Okay, thank you for the clarification. Okay, next question. Are low T regulatory cells an issue?
Yes, definitely. Yeah. T regulatory cells are also really important. I probably didn't emphasize it enough at the presentation, but they are definitely really important for these whole context, immunological context. So T regulatory cells, basically, what they do is send cytokines to promote the growth or the development of other immune players, such as, for example, natural killer cells or TH cells and other immunological players, other immunological cells. So definitely T regulatory cells are crucial for the establishment of fertility, placentation, and implantation. So low T regulatory cells are an issue. Well, as always, we're different people. We have different like starting points. So I cannot give you an answer just from this information. But low regulatory cells should be taken into account. Yeah, we should look at them more in depth. I hope this answered your question, Emily, and thank you.
Thank you so much. And Emily has added a follow-up. Is there treatment?
Yes, there are definitely treatments, but again, immune treatments have to be taken just looking at the whole thing, because I cannot give you I cannot say take these or take that, because then these treatments will also have an effect on other cell populations. Therefore, we'll have to look at the whole situation to provide a treatment. Yeah.
Thank you. Indeed, of course. And as you can see, thanks from Emily is right here.
Emily.
All right. There are like 2 questions left. Okay, let's have a look. Brittany has added one more. Could unnecessary intralipid therapy, utropin NK cells normal, negatively impact a transfer?
Well, this is a bit. Thank you for that question. It's a bit controversial because I am not a very. I don't support to give immune treatment like all together and like different situations in combination with other treatments, because they are complex, and we have to look at the effect that they do have in other cell populations. So I'm not sure if the therapy itself was the reason why the transfer didn't work in this case. I think, Brittany, you asked earlier if like a combination of different treatments could be dangerous. Well, I think we have to be careful about these combinations because sometimes they even have opposite effects. So definitely get in touch with a specialist, with an immunologist that will provide you more adequate answer and treatment to these situations. I hope this answers your question. No worries. Thank you, Brittany.
You can see in this comment, nothing else to add. Thank you so much, Brittany. And now I think we will have our final question for tonight. So let's have a look. I had 3 failed transfers, in total 4 quality embryos, none of them implanted. I am 31. Would you recommend to proceed with NK cell tests?
Well, definitely, yes, I would say. Well, thank you very much, Monica, for your question. Yeah, it looks like there's something else there that we are not seeing. So I definitely recommend to look more in depth. As I was saying, immunological problems are sometimes there, and we are not able to see them. So definitely encourage you to have a look at the natural killer population as well as the whole immunological context. There's sometimes easy answers to our issues, but we just have to look at the right places. So yeah, definitely encourage you to look more in depth, and please keep trying because I'm sure there'll be an answer.
Excellent. Thank you so much for that question indeed as well. And well, someone is typing. So as always, I just want to make sure whether we have another question or it's just a thank you, because I can honestly say right now that it's been a brilliant session with a lot of interesting questions. So thank you so much, everyone, for those. And I can only say that definitely it's, as you can see comments here, thank you for the talk. Okay, there's one more I need you to see. Thank you, Elena, for all the helpful information. Congrats on the presentation. Yes, agreed on that as well. So indeed, very, very useful. Thanks a lot, everyone, for joining us tonight. I do believe it has been helpful. I can see your thanks. So that means it is helpful. Sorry, one more question. I guess we can still answer, right, Elena? So
yeah, definitely. Yeah, more than
how long does the care blood testing take?
It's really quick. Well, yeah, it takes the time that you take the blood, and of course the time that it takes for us to receive it at the lab, and then after that, we will take no more than 15 days to give you the results. So yeah, those are our times, and of course it's pain free, and I think it's worth looking at it if you have several failures and you cannot find an answer. Yeah.
Okay. Thank you. Sorry, but one more. I still think we can answer, right? Sorry, one last question. Should my husband be tested for anything? I'm so sorry if you answered this.
No, it's okay. Well, as I was saying, it is important to look at everything. So if you are getting tested for KIR genotype, as I said earlier, it's okay. I'm happy to answer. If you ended up being KIR AA, then yeah, I'll encourage you to. I encourage your husband to get tested as well. Then we can find out about the whole possible genotype of the embryo, because testing the embryo is not possible at the moment. But yeah. So if you ended up being KIR AA, then yeah, your husband should be tested as well. However, you ended up being KIR BX, well, then maybe that is not the problem that is causing infertility. I hope this answers the question. Yeah, okay. Thank you, Brittany.
It definitely does, as you can see. So huge thanks, everyone. This has been, as I mentioned, brilliant session. Thank you for all your questions.
Yeah.
Elena, you have been excellent with answering all of those as well. So thanks a lot. And of course, I just want to mention that this has been recorded. So if anyone would like to have a look at this once again, or you would like to share it with anyone, you can go to our website, myivfenses.com. You will be able to find our previous webinars and, of course, 2 webinars with Elena. So thanks so much. And, of course, on our YouTube channel, it will be uploaded tomorrow as well. Elena, thank you so much. Before I let you go, anything else you would like to add?
Yeah, I'll just like to thank you and everyone watching tonight, and for all the questions. They were great, and of course, as I said last time, I'll be more than happy to answer more questions. If you have any questions, please get in touch with Caroline. And thank you, Caroline, again for inviting me.
Thank you. Thank you indeed. It's always a pleasure to have anyone from IVF Spain. You are always doing an amazing job. So thanks a lot. This has been your second webinar with us, and I'm already looking forward to more events with you, Elena, for sure. And again, thanks so much, everyone. Have a lovely evening or day wherever you are. And as you know, we will be back here tomorrow at 8 p.m. UK time. Another topic is right here with us. So I hope you will be able to join us. Thanks so much. Take care.
Bye.
Bye.
Bye.